Hello, everyone. Welcome to today's live broadcast, NGS and Clinical Research, presented by Jennifer Fostel. We are excited to bring you this educational web seminar presented by LabRoots and sponsored by Qiagen. As the innovative market and technology leader, Qiagen creates sample-to-insight technologies that enable access to valuable molecular insights from any biological sample. The mission is to make improvements in life possible by enabling our customers to achieve outstanding success and breakthroughs in life sciences, applied testing, pharma, and molecular diagnostics. To learn more about Qiagen, please visit www.qiagen.com/US. I am Brenda Kelly-Kim of LabRoots, and I'll be your moderator for today's event. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time during the presentation. Just click on the green Q&A button located in the lower left of the presentation window. Type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. Finally, if you have any trouble hearing or seeing this presentation, please click on the support button at the top right of the presentation window to let us know. Or you can use the Q&A button and we'll try to resolve your problem. This presentation has been approved for continuing educational credits. Please click on the Get Your Free CE/CME Credits button at the bottom left corner. This will take you to a page listing all of our speakers and presentations. Please select the CE/CME button under your presentation and follow the instructions to claim your certificate. I'd now like to introduce our presenter, Jennifer Fostel. Jennifer has her master's in molecular and cell biology from the University of California at Berkeley. She did her graduate research in Dr. Susan Marques's lab, where she analyzed the key properties of proteins from thermophilic and mesophilic microbial species. She has worked in next-generation sequencing for the last 7 years, including management of clinical next-generation sequencing projects for the Broad Institute of MIT and Harvard, as well as NGS biomarker assay development for Takehome Pharmaceuticals Oncology Translational Medicine. Today at Qiagen, Jennifer is a Senior Global Product Manager covering a portfolio of universal workflow NGS library preparation products. Please join me in welcoming Jennifer, and I now turn the presentation over to her.
All right, thanks, Brenda. So hello, my name is Jennifer Fostel, and I'm a Senior Global Product Manager for Qiagen's NGS Life Sciences Group. And I'd like to thank everyone on the call for joining us for this webinar today. So I currently work in the development of new next-generation sequencing wet lab products for Qiagen. And as NGS has become more common in the clinical environment, I found I frequently run into clinicians and clinical researchers at events such as this one who are interested in exploring new techniques for their research, but aren't quite sure exactly what makes next-generation sequencing a different type of test from standard Sanger sequencing, or how clinical validation of an NGS assay might be similar or different from other commonly used biomarker discovery techniques, such as IHC or qPCR. And yet every year, there's sort of an increasing number of next-generation sequencing-based assays that are validated and enter into clinical practice, or are developed for preclinical research purposes. NGS being used in the clinic today includes pre-implantation genetic screening, liquid biopsy detection of tumor mutational profiles, sequence-driven chemotherapy decisions, as well as determination of biomarkers for response to a particular drug or treatment regimen. So shown here on the slide, I've included just a few examples of ways in which next-generation sequencing is starting to become common practice in a clinical environment. So on the far left is a very recent Newsweek article, I think from just a month or two ago, that describes ways in which researchers at Children's Mercy Hospital in Missouri, along with Genome, a company in California, they've worked together to address one of the major challenges associated with implementing NGS in the clinic. And that is a historically much longer turnaround time than typical clinical diagnostic tests from the time that a sample is collected from the patient to the time that that result can be returned to the physician to influence patient care. In the center is a paper from industry test provider Foundation Medicine. This paper basically describes how they went about developing and validating a clinical grade NGS test specifically for the oncology market. And then on the far right is an investigator paper published by researchers at the Dana-Farber Cancer Institute, in which human whole exome sequencing along with computational clinical interpretation was used to actually guide patients' precision cancer care in real time. And so to start off this presentation and start to talk about, how researchers get started doing this type of work, or, what is really meant by next generation sequencing, I'll go through a few basic agenda items. So first, I'll set the stage by providing some background on exactly what is meant by a next generation sequencing technology. And additionally, I'll walk through some main types of NGS assays and provide some discussion on why a particular type of assay might be a good fit for a study or not. Next, we'll talk about some of the key questions that a researcher may want to explore when deciding, to implement an NGS assay in clinical studies for the first time. And finally, I'll try to get into a few specifics about how to validate an NGS assay for use in a clinical environment. And the example that I'll use is Amplicon panel-based targeted resequencing. This is a relatively common NGS assay type for clinical use. And I'll kind of use that example to talk about some of the CLIA guidelines that can be followed when implementing an NGS assay, as well as kind of go through some hypothetical data of what validation experiments and validation data for an Amplicon panel type approach might look like. Some things that I am not going to include in today's talk. Number one, there is a ton of change and progress going on in the regulatory environment for clinical NGS. But I am not a regulatory expert, and so I will leave this to other speakers at this event to fill in details about the current thinking and the current movement from FDA, as well as some of the other clinical advisory groups looking at next generation sequencing. Second, one of the major challenges for implementing NGS in the clinic continues to be the design, implementation, and standardization of bioinformatics pipelines to analyze the massive quantity of data that is produced. This could fill an entire talk on its own, and so I'll kind of leave this as an exercise for another talk or another time to go through some of the basic ways that NGS data can be analyzed. And then finally, the last topic that is very interesting, but I think doesn't fit in today, is reporting standards for clinical NGS data. So at the end of the day, what we're looking for in a clinical test environment is sort of actionable test results that a physician can use, or that a clinical study can use to draw conclusions from the data that are produced. There's still a lot of debate in the field and a lot of different ideas about how to do reporting for clinical NGS. And again, I'll leave that for a different talk. All right, so getting into an introduction to next generation sequencing. So what makes something, or what makes a sequencing method grouped into this category of next generation or next generation sequencing? So NGS comprises a collection of relatively new technologies or new techniques that have really been commercialized in the last decade or so to allow rapid sequencing of large portions of an individual's DNA or RNA, or even their entire genome. And this has been made possible largely by techniques that allowed a rapid decrease in the cost per base of the individual sequencing reaction, largely through miniaturization of the assay, automation of the chemistry, and automation of the analysis or base calling portions of the workflow. And so to kind of illustrate this, I like to show it as two basic technology jumps that many people listening to this webinar may be familiar with. So the first is, I don't know how many people listening have ever run a sequencing slab gel. I personally have run just one. I would not do it again. So this is the way that sequencing was done in a basic research environment until probably 10 to 15 years ago. And so the idea is basically that with this huge slab gel-based apparatus, using radioactive nucleotides to produce banding on an autoradiogram, you could run just a few samples per gel, up to about a 300 base pair max read length if the quality of data was really good. And base calling was actually done visually by looking at the pattern of the different bands on the gel. And so the throughput of doing this, like if you can imagine trying to sequence a whole genome using these gels, it would really take you forever and it would be extremely tedious, and there was no good way to automate it. So that first major technological jump was the switch from gels to sequencing using capillary electrophoresis. And so now instead of separating samples by running different lanes on the gel, the samples were actually separated into their own individual, very fine glass capillaries. And so what this did was it allowed a higher number of samples per run, between 8 and 384, depending on the version of the technology. Longer reads because that thin capillary had better resolution. And also instrumentation was developed to facilitate automated loading and automated base calling of those sequencing reactions. So the next jump then from capillary to next generation sequencing follows that same pattern. So what we're going to do again is miniaturize that sequencing reaction even further. So instead of a small bundle of capillary tubes that you can hold in your hand, we're actually transitioning to a microscopic 2D array on a surface. But the same idea, right? So we have individual sequencing reactions. Now, instead of each one having their own capillary, those sequencing reactions are bound to a solid surface and separated in 2D space from each other. And so this allowed us to jump from hundreds of samples or dozens or hundreds of samples from capillary to even the 2007 original Solexa sequencer, which could sequence 40 million simultaneous sequencing reactions on a glass flow cell about the size of a microscope slide. And so the key here is to separate those sequencing reactions in physical 2D space. And there have been a couple of different strategies for doing this. The top picture on the right there is of the original 454 technology, in which DNA is amplified on tiny beads, and then those beads are settled into wells on the surface that keep them separated from each other so that they can be imaged. And then on the Solexa flow cell at the bottom, there's actually a lawn of primers coating that glass surface, and the samples are covalently bound to those primers, allowing them to stay put and stay in one place so that they can be imaged through the course of the sequencing reaction. And again, the point of all of these technologies is that by making the sequencing reaction smaller and being able to run a larger number of simultaneous reactions, or people will say massively multiplex, this caused a massive drop in the cost to sequence a human whole genome. And by reducing the cost per base, we've actually been able to expand the number of types of studies and the number of types of questions that were economically feasible for researchers to address. Okay, and so then the next slide, I'd just like to show a few of the different next generation sequencing technologies that have launched over the last 10 years. Starting with 454, which is a pyrosequencing based technology in the mid-2000s. Helicose, which was one of the first single molecule sequencers. The Solexa or Illumina-based technology in the late 2000s. It was a sequencing-based synthesis technology still used in the HiSeq and MiSeq instruments today. Solid, which was a next-generation sequencing by ligation technology. Pacific Biosciences, which is a little bit of a different technology. It's a long read chemistry, where the sequencing is done by individual polymerase molecules held in a tiny well with a single molecule of DNA circulating through that polymerase. The ion torrent instruments at the top there, these are, instead of taking pictures of the sequencing reaction and relying on fluorescent nucleotides like the Illumina instruments, the ion torrent instruments actually detect the hydrogen ion that is released through incorporation of a nucleotide to the sample DNA. And then just recently at AMP last week, QIAGEN also announced the latest NGS instrument, which is the Gene Reader NGS system. And again, this is sort of an Amplicon-based sequencing technology that is combined with end-to-end support so that the instrument includes sort of your sample prep, your sequencing, as well as the analysis steps of the workflow. Okay, so I wanted to next talk about some of the different types of NGS assay types, because when getting into utilizing NGS in a clinical study or in clinical care, there are a lot of questions about what assay would be most appropriate for the question that is being asked. So NGS assays are unique among many testing methodologies in the sheer amount of data that can be generated, and the lack of an a priori definition or hypothesis of what will be examined. Also, the number of clinical interpretations that can be made from a single sample. This is really one of the prime benefits of doing NGS for clinical studies. You just, from the same sample that could be collected from a patient, you're able to run thousands, even millions of genetic tests simultaneously. And any one of those tests could be the one that has utility to diagnose or predict an individual's risk of disease. Okay, so just to walk through this slide and explain what we're looking at. So on the left, whole genome sequencing is sort of the largest region of interest assay that you can do. This essentially involves no selection of particular regions of the genome or particular genes of interest. This involves sort of shotgun sequencing of all genomic material present in the patient sample. Whole exome is the next one over from that. Reasons to do whole exome, so essentially people will do this because it still allows you to cover all of the protein coding regions of the genome. But in comparison to sort of the 3 gigabase human whole genome, an exome covers about 40 megabases. So as you can imagine, if you have a limited or restricted budget, you're going to be able to do a lot more human whole exomes for the same amount of money that you would be able to do whole genomes. In the middle there is PCR Amplicon-based sequencing. This is a very popular approach, in part because it allows you to be very selective about the regions of the genome that you are querying through your test. So in Amplicon-based sequencing, you have carefully designed primer pairs that will amplify up for sequencing only the genes or the portions of the genome that you're interested in. And this, you can design a very small region of interest that again allows you to sequence a larger number of samples for your study or compare a larger number of patients with the same diagnosis than you could if you were looking at a larger region of interest test. Other options on the right that I won't talk about too much in this talk include transcriptome sequencing or RNA-seq. Again, extremely powerful method using the RNAs derived from a tissue sample, which in addition to sort of variant calling and mutation identification, RNA-seq can allow you to look at gene expression, gene fusions or structural rearrangements, as well as splice variants that you would only be able to tell from sequencing RNA. And so the key here is really to look at the region of interest that your assay is going to explore. So the larger the region of interest, the greater your data storage and analysis costs will be, but the more general your assay is. So if you have multiple clinical trials or you expect to be sequencing patients enrolled over a long period of time, having a more general study or assay design will allow you to combine those patients together and actually analyze them as a set for greater statistical power. On the other side, the smaller targeted panels are easier and less expensive to run for a given project. But if you have sort of 5 different clinical studies running and each of them is running a unique and non-overlapping set of Amplicon targets, it's going to be much harder for you to combine those data sets together and actually analyze them at a larger scale. Okay, and this just systematizes those applications again and shows kind of, the idea is basically that, and sorry, that should say frozen samples and frozen tissue and FFPE-based tissue. You know, sort of the highest number of genes or the highest coverage of the genome would be whole genome sequencing. A typical depth of affordable sequencing for WGS would be maybe 10 to 60x coverage. That means each base in the whole genome would be covered by the sequencing reaction at least 10 and up to about 60 times for accuracy. In the middle, you have kind of your exome or RNA-seq. And for exome, a typical coverage is often about 100x. And then there at the bottom, you have custom Amplicon panels or custom capture panels. And because of the small region of interest, you're able to drive the sequencing depth to a much higher level, potentially 1,000x or more. And so particularly oncology researchers tend to like doing this because let's say that your variant of interest is present in a very small fraction of your tumor sample or biopsy. You're going to have greater sensitivity to detect that with the higher depth coverage at an amplicon or exome level as compared to a whole genome level. And again, the NGS advantage for clinical research for all of these applications is that from the same sample that previously may have been submitted for just a few qPCR tests, you're now able to obtain diverse genomic information. Okay, so let's assume that you have a clinical study coming up and you're interested in running an NGS assay. I wanted to include a few practical considerations for some of the next kinds of things you may want to think about in moving forward with your testing plan. So first of all, NGS workflows generally require a library prep step. And so what do I mean by library prep? So you collect your sample, you extract nucleic acids. In this case, we'll use DNA as an example. And so right there in the middle, you can see there's sort of a cartoon representation of double-stranded sample DNA. And for most short-read technologies, this would additionally be fragmented to approximately 200 to 600 base pairs to be compatible with the surface binding chemistry of either ion torrent or Illumina-based chemistries. And so the next step is to add adapters. And it's once those platform-specific adapters are added that it's considered an NGS library. For Illumina and ion torrent, that is generally a linear design, so known sequence adapters are ligated onto the ends. And PacBio, because they have more of a continuous or circular loop of DNA going through the polymerase, they actually add adapters in the form of what they call the smart bell. So it's just a little hairpin loop that goes on the end, but otherwise functions similarly to the Illumina and ion torrent adapters. And so one of the things to consider is how much DNA is required for NGS library prep, and how does that compare to the amount of DNA that you can get from the sample type available from your study. So historically, standard NGS library prep input has been about 100 nanograms to 1,000 nanograms, right? So about 1/10 of a microgram up to 1 microgram. And that is very easy to get from a human blood sample. 200 microliters of blood will generally give you, between 5 and 6 micrograms of DNA. But for other sample types, for example, single cells, bacterial cells, circulating DNA isolated from blood plasma, getting up to 100 nanograms can be really, really difficult. And so there's been continuous drive to reduce the amount of DNA required for NGS library prep and sort of enable some of these additional applications. So right now, there are certainly library prep chemistries that can get down into the 1 nanogram range, which is practical for, as I mentioned, cfDNA isolated from plasma. And then getting even lower than that, single cell sequencing from both mammalian and bacterial cells is also possible through whole genome amplification technologies, such as molecular displacement amplification. The next thing to think about when assessing the sample type available for a study is how much DNA you'll be able to get reliably from your sample type, and whether that is enough for the question that you want to ask. And this is particularly important for heterogeneous samples, such as tumors, where you actually may have many different subclones present in the tumor that each have different variants or different mutational profiles that you want to analyze. So some quick math that it's helpful to do just to sanity check the study design is how much genomic diversity are you going to have in the amount of DNA that you can reliably extract from your samples. So assuming that, and again, we're going to use a cfDNA example for this just to make the math a bit easier, but assuming that there are about 6 picograms of DNA per human cell, and that all parts of the genome are equally likely to be represented in DNA extracted from your sample. And for cfDNA, you can assume that the DNA isolated from plasma is actually mostly derived from the tumor. So between 80 and 100% tumor-derived, just because tumor cells are slightly more likely to shed DNA than standard, lining of your blood vessels or other cell types. So essentially that means that you have a fixed amount of genomic diversity present in the number of picograms or nanograms of DNA that you can isolate from a sample. And so you can see that if you can isolate approximately 1 nanogram of DNA, your theoretical maximum unique sequence depth, right? So sequencing depth derived from unique molecules of DNA present in your sample is about 350x. And so fortunately, that is a very reasonable level. So many successfully extracted cell-free DNA samples for 5 to 10 mils of plasma can definitely get into the 10 to 100 nanogram range. And if you're lucky enough to be sequencing from human whole blood or a frozen tissue sample or even sufficient slides of FFPE, again, you're going to have no problem sort of hitting that range. Another thing to take a look at, however, is that 350x is sort of a best case scenario. And in reality, the DNA that you're able to extract from a sample does not all make it onto the NGS flow cell for conversion into data. And so this is just a sample loss calculation using, for example, the Illumina TruSeq custom Amplicon protocol. This is a very common Amplicon-based targeted resequencing protocol, but it's quite a long protocol and it involves a lot of different steps. And as a result, there's considerable sample complexity loss through each of those processing steps. So you can see here that just through basic molecular biology, binding to the filter, doing PCR, binding to Ampere spry beads for buffer exchange, denaturing the sample, and finally loading it onto the flow cell, what you end up with is really a very small fraction of even the input DNA that began the NGS library prep process. And so assuming that now we've only got about 0.2 nanograms of DNA from our original hundreds that are going to make it onto that flow cell to generate data, our real-world maximum unique sequencing depth drops rapidly from about 350x to about 70x. So that's still plenty for many applications. Obviously, for any kind of Mendelian sequencing application, you're going to be looking at Mendelian homozygous and heterozygous variants. There's no issues with trying to call those variants from a 70x threshold, but for tumor sequencing, it's important to try and preserve the genomic complexity of the sample as best as you can through the selection of your library prep process and the elimination of any of those processing steps that aren't truly necessary. So the next challenge for establishing NGS in the clinical environment is following sort of the clinical guidelines specifically for doing testing in a clinical environment. So in the last 2 years, guidelines for the standardization of clinical NGS assays have started to emerge, including an NGS checklist from the College of American Pathologists, the standardization of clinical testing work group, the American College of Medical Geneticists guidelines for NGS, and the Clinical Laboratory Standards Institute's guidance on best practices for the development of clinical grade NGS. In addition, there's been the approval of 2 510K approved NGS instruments by the FDA. And this has provided some of the first insights into how the FDA views NGS-based tests, as well as the growing expectation that NGS assays for clinical use should be standardized and validated just like any other clinical assay. And so, as historically, facilities and providers who have strong expertise in NGS have not been clinical laboratories, that brings up some questions about how do we merge the necessary technical know-how for these technologies with an understanding of the regulatory environment and how to operate under a clinical grade quality system. And so that brings up the next big question, which is, you've decided to incorporate NGS into a clinical study. You've identified the application that would be the best fit, and you're sort of aware of the clinical guidelines for validation. So the next step is, do you try to do this within your organization, or do you outsource and look to a service provider or a testing lab that is outside and experienced in running NGS? So some reasons to insource you have access to significantly lower turnaround times. So if you have a study where it's really important to get data back so that they can influence patient care in a more or less real-time basis, shipping samples a long distance to a third-party provider, allowing them time to run their assay, prepare deliverables, get them back to you, is generally going to be longer. And so the more of the process that you can do within your own control or within your own organization tends to bring down that turnaround time. When insourcing or using your own clinical lab, it's also much easier to link the NGS data with the clinical outcomes information that allows that data to be interpretable. You also obviously have full control of the laboratory process, which can make the publication process much easier. And as we mentioned, a reduced number of wet lab handling steps, so you don't need to transfer sample into a fresh tube to send off for the testing lab. And fewer QC steps. I mean, you're probably going to QC the sample before it leaves your facility. The service provider is going to QC the sample again when it arrives. Starting to eliminate some of those steps will leave more DNA or RNA available for the assay. But obviously common challenges, trying to run NGS assays in-house does require significant investment in expertise, trained staff, equipment, and compliant process development. On the other side, reasons to outsource the assay. So being able to access the cost and benefits of a facility that do NGS regularly and at scale. This is a big one and difficult for labs that are just going to do a small amount of NGS testing to replicate. Higher throughput labs are also more likely to offer automated processes that have higher assay reproducibility, which makes interpretation of the data significantly easier. And it is often easier to select a qualified vendor than to physically build all of that infrastructure yourself. On the other hand, common challenges. As I mentioned, vendors who are experienced in both clinical trials and NGS are quite limited. And so this means that there can be significant coordination and education in both directions required by the investigator or study sponsor. This is just to give a picture of, some of the organizational complexity that can become the reality for setting up some of these assays, especially in the context of clinical trials. So imagine, you have your study sponsor, usually contracting with a CRO or a central lab running the study. You have your clinical sites where the patient samples are actually being collected or the blood draws are happening. Maybe you have an NGS vendor you're contracting with to do the wet lab work, but an analysis vendor who will actually be then processing those data and turning it into an interpretable, understandable conclusion. And so this all can create quite long processes that take a lot of time and require a lot of project management and coordination. And so it's good to be aware of this potential complexity before getting started, so you can make good decisions about how much to do in-house, how much to send out, and potentially whether you will need staff on hand to manage some of the transfers between organizations. So here I include some sample questions. If you decide to evaluate NGS vendors and you want to kind of get a sense for how they stack up against each other and which one you may want to work with in the future. So number one, by far, is the quality of the data they produce. So not all NGS data is created equal. You're going to want to be very clear on what kinds of reports they're able to provide you with. Are there in-process reports or just a report at the end? What kind of data quality metrics will you receive? What are their data quality guarantees, and what happens if the data that they produce do not meet those guarantees? As well as how they're validating their assays, so that they can guarantee to you that the data quality metrics that you're being provided with were established in a validated way. Project experience is a huge one. So does the vendor that you're exploring have previous experience with projects of a comparable scope? Have they worked with your sample type before? Are they experts in your sequencing assay, or have they only really specialized in one type of assay? And then finally, what are their sample requirements? In order to do an Amplicon-based test or human whole genome sequencing, for example, how much sample is required, how much DNA, how much RNA, If it's tissue, how much tissue do they require? Technology available. There's a lot of different kinds of equipment and laboratory materials that are needed to run some of these tests. So if you don't want to kind of partner with a vendor and build up that process from scratch, it helps to make sure that all of that equipment and technology is available and pre-validated before your study begins. Assay transfer experience is also one. So if you plan to develop an assay in your own lab and then transfer it over to an NGS service provider, how much experience does that vendor have with taking in an externally developed assay and implementing it in their lab? Bioinformatics capabilities, so don't be afraid to work with separate wet lab and analysis vendors. It's relatively rare for a vendor to be good at both. What turnaround time can they guarantee or offer for your project, and what kind of deliverables are you going to get in that time frame? What quality processes are in place, and particularly if you plan to use your NGS assays for treating patients for standard of care or for anything beyond an exploratory endpoint for a clinical trial. You're going to want to make sure that the facility is CLIA or CAP accredited, at least in the U.S. And then finally, one that I like to put in that maybe doesn't always make it to the list is the facility's customer service and responsiveness. NGS assays are still relatively expensive and require a large commitment of very precious samples that are difficult to obtain. So you're going to make sure that you are able to get the information and the feedback from the vendor you select in a timely fashion. Another thing to watch out for is to make sure that the vendor is extremely comfortable with the key parameters for validating a clinical assay. And so these are sort of the CLIA guidelines: standard, accuracy, specificity, precision, reportable range, sensitivity, and reference range. And so you're going to want to make sure that either your internal clinical lab or an external provider is very comfortable interpreting these parameters in the context of an NGS assay. So for example, just to talk about for accuracy, they should have parameters for filtering out low quality data. They should have standards where they will not return test results unless there is adequate sequence depth and uniformity of coverage to support the variant calls that they are making. They should have a procedure for confirming any clinically actionable variants through an orthologous technique, either Sanger sequencing or qPCR or something else. For reportable range, this one is interesting. So reportable range for NGS, one way that I like to look at it is this is the region of the genome that the test is capable of testing, so or is capable of querying. So for example, if you have an Amplicon sequencing design where you're really just looking at a small number of genes or a small number of genomic regions, you're going to want to see that all of those regions have been validated and the test is capable of delivering high quality, sufficient depth data for all of those regions. And if not, there are difficult parts of the genome, repeat regions, certain alleles that just don't amplify well. Those should really be eliminated from the reportable range for the test. Sensitivity, so this is particularly important in the context of oncology sequencing. So what is the lowest fraction of the sample that could contain an oncogenic variant of interest that the assay would still be able to detect? And then finally, a reference range. So ideally, we would have a reference range with all the possible outcomes from a clinical test. But currently, we are not to the point where the human reference genome includes every possible variant from every possible person who could be studied. And so this doesn't really exist for NGS today. In the future, we hope that there will be population-specific or disease-specific references that will serve as a more complete reference for clinical NGS. But for the time being, reference range is one area in which NGS assays probably will continue to be a little bit different from many other clinical tests. Okay, so for the last part of the talk, I'd like to go through just a few examples of what a real-world clinical assay validation might look like, specifically for an Amplicon targeted resequencing panel. And just to give you an idea of some of the experiments that might be done, the designs of some of the experiments, and what some of those data might look like. Okay, so here is a hypothetical assay design This will be a Amplicon-based panel with 1,000 Amplicons covering 50 unique genes and a total region of interest of about 100 kb. So this is relatively small. Again, I mentioned that the human whole exome is about 40 megabases. So this Amplicon design is really a very small fraction of a total exome or whole genome type sequence. Our Amplicons will be about 160 base pairs because we plan to use it with a cell-free plasma-derived DNA sample. And cell-free DNA tends to be relatively fragmented, about 200 base pairs to start. So we wanted to make sure that our Amplicon length was slightly smaller than that. We are isolating our DNA using the Qiagen Circulating Nucleic Acids Kit. Our library prep, I've just put in here the Qiagen One-Step Amplicon Library Prep Kit. This will be a new product coming out in December. It will be specific for Amplicon sequencing, so it fits well in this workflow. And then for the sequencer, we're going to choose an Illumina MiSeq for this experiment at 2 by 150 base pair sequencing. And the next thing we're doing is just checking on the sequencing depth that we can generate. So assuming that our MiSeq run can yield us about 5 gigabases of sequence, if we divide that out across 100 kb of region of interest, as well as 96 multiplex samples together per flow cell, it means we're targeting about 500x mean target depth. And there at the bottom, I just have a quick diagram to show the difference between how this experiment would look if it were a hybrid capture targeted resequencing experiment versus an Amplicon-based targeting resequencing experiment. So imagine that these lines are sequencing reads that are being aligned to the human reference genome across the top. You can see that the hybrid capture data tends to be more staggered. Those reads have random start and stop points, whereas the Amplicon data, your reads will be neatly defined by the primers in your Amplicon panel design. And so there are some pros and some cons to this, but it can make the analysis of the data a little bit more straightforward, just because you know exactly which regions you're trying to amplify with each of your primer pairs. Okay, so some of the experiments that we would run to validate this Amplicon panel include looking at the accuracy and the specificity. So we have multiple cell lines with known cosmic mutations, preferably pre-verified via Sanger sequencing or qPCR. And these can be basically, we can run our panel using DNA extracted from these cell lines and make sure that we are detecting all of the variants that we expect to see from those sample types. Reportable range, I mentioned, we need to look at, okay, let's assume that we're going to have samples with a DNA input between about 25 nanograms and 150 nanograms. So at those levels of input, are we able to get sufficient sequencing depth for all of the Amplicons in our panel design? So sensitivity or limit of detection. This is an oncology-based assay, so we want to do a titration, basically, of cell line DNA with a variant of interest into variant-free wild-type DNA. And by titrating it down between 105 or 2 or 1%, we're able to actually see how low the assay can go and still detect that variant. And then finally, we always want to assess assay performance using a realistic sample type. So since our clinical samples will not be cell line DNA, we want to make sure to qualify the panel using real-world cfDNA samples as well. All right, so one of the first things that we want to take a look at is our ability to generate high-quality sequence data that meets the manufacturer's specifications. So we never want to be trying to interpret clinical NGS data that doesn't match the specifications that the NGS manufacturer says their platform is capable of generating. And so here we have a titration of different input amounts and samples run in duplicate. And we want to make sure that we're seeing very high quality sequence. So that's shown in the percent greater than Q30 column. So we like to see the percent greater than Q30, at least 80 or 85%. And all of these are nicely above that. And then, excuse me, in the column next to that, we have the percent mapped. There are typically things like contaminating DNA or sequence reads derived from the adapter sequence, and those are not going to map to the human reference because they're not human sequences. And so we also like to see that the percent mapped stays nice and high, that our process is working well, and that the sequence that we're generating really is mappable human DNA sequence. Another example of some validation data, this is, again, taking a look at the reportable range. So this is actually separating out the data that we've generated by the gene or the amplicon or the target region. So in this example, they're all different genes. And so even though we drive the whole experiment to an average of 400 or 500x steps, the actual coverage for each gene included in that panel may be slightly different. And this may be because that allele is a little bit more difficult for a polymerase to amplify. The design of the primers for that gene may have been more challenging. And so what we do is we kind of break it down and we make sure that there aren't any genes that the panel is having reproducible trouble generating coverage for. And so in this panel, you can see there's kind of one high outlier, a whole bunch of genes that are coming up with sort of an average or middle sequence depth, and there's just a couple of genes there at the bottom that the panel is having a lot of trouble with. And so for a clinical assay, it would be very unlikely that you would be able to get the kind of sequence depth that you're looking for interpretable variants from these genes. The recommendation would be then to remove these from the assay reportable range. On the right-hand side, reproducibility of target coverage. Again, just taking a look at the range of nucleic acid inputs to the assay and making sure that you are seeing approximately equivalent performance for each gene of interest across that range. Another potential experiment is looking at the reproducibility of somatic allele frequencies. So again, if we're sequencing cell line DNA, we would expect to be able to call a homozygous variant at close to 1.0 frequency amongst the sequence reads, whereas a heterozygous variant, you'd want to see approximately 50% of the reads supporting that variant call across all of your samples and across all of your potential input ranges. And we see that nicely here. Another key experiment to do, particularly for oncology, is that assay sensitivity or limit of detection. One common way of determining the limit of detection is just to do a mixture experiment where you have cell line DNA with some known mutation of interest, and you actually mix that in different ratios with a wild type DNA that does not include those variants. And then what you're hoping to see there in the figure at the bottom is a nice linear correlation between the observed allele frequency on the y-axis and the theoretical allele frequency based on your mixture experiment on the x-axis. And so in this case, you'll often see two populations. The ones that are, approximately linear at about y equals x are your homozygous variants, and then I have variant C set up as an example of what a heterozygous variant might look like, where even at 100% cell line DNA, it's really only getting up to about an allele frequency of 0.5. These are all hypothetical data just to give you an idea of what it'll look like. Real world data can look quite different and will vary significantly based on the variant itself. So some variants, you'll have much higher sensitivity. Variants that are difficult for the assay to amplify may show unusually low accuracy. Unusually low sensitivity. And so what many researchers will do is they will, number one, average all of their test variants together to get an overall limit of detection or sensitivity figure for the assay. And then also if there are key variants, like you really want to be able to look at EGFR or you really want to be able to look at a particular KRAS variant, you may calculate a specific limit of detection for those variants to make sure that the assay is capable of giving you the key data that you're looking for. And then finally, I wanted to mention how important it is to always validate assay performance with matched sample material. So if you know that your clinical assay will be done on FFPE tissue or cell-free plasma-derived DNA, be sure not to validate your assay exclusively using cell line DNA. You can source clinical or commercially available samples that are the same type as your clinical material to be tested, and you're going to want to ensure that the assay still meets all of those data quality standards established by your validation experiments. So some limitations of NGS as a clinical assay to start to wrap up. So again, NGS, as you've probably noticed, it does require significant setup costs. There is specialized instrumentation, particular training that the staff require, and the implementation of an analysis or interpretation solution that a lab that's not already doing NGS is unlikely to have access to. Also, the majority of technologies being applied to the clinical environment are short-read technologies, and those are not particularly well suited for identifying large structural rearrangements or insertions in the genome. So, for example, short tandem repeats, common in sort of forensic human identification tests, internal tandem repeats, such as what's found in the FLT3 gene, anything that changes the fragment size of the Amplicon is going to be difficult to assess with a short-read technology. Another concern is that many common clinical sample types can be difficult to work with for NGS. So, for example, fixed bone marrow aspirate clots or anything that has a very high cross-linked protein content can be difficult to extract DNA and RNA from. And as well as samples where it's difficult to get a particularly pure tumor biopsy. So a very high percentage of the sample containing the normal genome of the patient will reduce your sensitivity to detect tumor variants. And another thing is that negative results, which is essentially to say no variant or association found, can be difficult to interpret. It's hard to tell whether there's really no variant present or if the assay sensitivity was just not sufficient to detect it. If the variant was present in the patient, but maybe not in the sample that was obtained. Or in the case of disease progression, was the sample too old to be relevant to the current clinical situation? And then finally, the clinical patient outcomes information necessary to interpret the data and the significance of the results can sometimes be harder to obtain than the NGS data. We can create standards for the quality of the NGS data, but in many cases there are not standards for the ways that physicians are reporting on patient phenotype or outcomes or the way that a patient is presenting. And so this can actually make it significantly harder to do any sort of statistical analysis that leads to clear conclusions from an NGS experiment. So just a few take-home messages then, now that we've sort of covered the intro, we've talked about getting started with NGS, and we've walked through a few examples of some validation experiments. So NGS basically comprises multiple new technologies that allow the rapid sequencing of much larger amounts of genomic information than most other standard clinical assays. At the same time, reliable and accurate NGS is definitely accelerating the development of personalized or precision medicine and has particularly valuable applications in inherited disease and oncology. But we should keep in mind that not all NGS data is created equal. Assay data and service provider labs should be held to strict quality standards, and all NGS assays used in the clinic should be fully validated according to CLIA guidelines. And the final thing is just to carefully consider the question being asked. Ensure that the samples that you're able to collect in a clinical environment have the power to answer the question being asked before beginning an NGS-based study. Okay, and just in conclusion, I'd like to say that NGS technology is clearly integral to the future of personalized medicine. And whatever regulatory approach is eventually adopted and the types of assays that start to become routine in the clinic, I think we'll all expect to see a great deal of continued innovation in this field as technologies and assay developments contribute to the advancement of medical care. And with that, I'll just say that quick plug for QIAGEN's portfolio of NGS samples insight solutions. So we certainly are interested in partnering with researchers who are starting to implement some of these technological methods. And we have a full portfolio of products available, from nucleic acid isolation through NGS library prep, as well as sequencing analysis and interpretation. And we encourage you to kind of check out our portfolio and see what we have to offer your research. And with that, I'd like to thank you for listening and take any questions.
Thank you, Jennifer, for that informative presentation. It is time for Q&A. If you have a question you'd like to ask Jennifer, please do so now. Click on the green Q&A button at the lower left of your presentation window, type your question into the box that appears on your screen, and click send. We'll answer as many of your questions as we can. And we do have a question already in our queue. Audience member asks, what is the difference between hybrid capture and Amplicon sequencing? Why might you choose one over the other?
It's a great question. Okay, so I can go back and I will show this slide really quickly. So hybrid capture and Amplicon-based data are two different ways to reduce the region of interest and sort of tailor your assay to just the sequences that you're interested in interpreting. The difference is that in Amplicon data generation, you're using PCR primers to only amplify up the sequences of interest. In a hybrid capture experiment, you're actually using probes, so nucleic acid probes, that are complementary to the sequences of interest, and doing like a pull-down experiment where you are capturing just the sequences you want to analyze through NGS and washing away and disposing of the rest of the genome. And so the reasons why you might do one or the other, Amplicon has an upper limit on how many different targets you can realistically amplify in the same tube. And so if you want more than about 100 kb or 200 kb worth of genomic regions to be included in your assay, you're more likely to go with a hybrid capture method. Likewise, if you want something that is going to facilitate easy analysis and straightforward interpretation, sometimes Amplicon-based methods can be a little bit easier to work with. And they're also easier to use with very small amounts of input DNA, which is a common problem in clinical sequencing.
Great, thank you. We do have another question. How can the QIAGEN gene reader instrument be used in clinical sequencing?
So the QIAGEN gene reader was just announced last week at the AMP conference in Texas. And the gene reader is a little bit different from other NGS technologies on the market in that QIAGEN is marketing sort of an end-to-end product. So instead of there being a library prep kit and a sequencer and analysis software, the gene reader NGS system is kind of all of those things all in one, with the goal of providing actionable results or actionable insights that clinical researchers can use, as opposed to requiring the piecemeal assembly of a workflow and figuring out which products to match with which. The gene reader system, I think, is really different from other NGS technologies in that it is targeted towards a clinical environment specifically.
Thank you for that. I think we have time for one more question. We do have it in here in the queue. Audience member asks, I am seeing Chiastic FX here in the NGS workflow. What is that?
So let's see, going back to the portfolio slide. So you can see under our sort of a workflow at the top where it starts from a sample, goes through nucleic acid isolation, library preparation and QC, and then sequencing and analysis. Chiastic FX is sort of there at the top. I think that's what they're asking about. This is a new library prep technology that QIAGEN has just launched specifically for whole genome sequencing and hybrid capture technologies. And so what this is an enzymatic fragmentation technology that generates those short read technology capable fragments that you can then either just sequence all of them and do a whole genome assay, or you can use hybrid capture-based targeted resequencing to reduce that genomic complexity to a region of interest and then sequence that. So Chiastic FX is our first library kit that incorporates this sort of enzymatic shearing workflow. And our performance data shows that it's quite comparable to Covaris, which is another technology that many labs use. but you have the benefit of it all sort of being able to be done in a standard thermocycler.
Great. Thanks for that answer. We are done with questions, but I'd just like to ask Jennifer Fostel for any last comments or advice for our audience members.
I think I've provided all the advice that I have for today, but thanks to all of the attendees for listening in, and I hope this has been useful.
Thank you, Jennifer, again, for being part of this virtual event. I'd also like to thank our sponsor, Qiagen, for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through May of 2016. You'll receive an email from LabRoots letting you know when this webcast will be available for replay, and we would invite you to share that announcement with your colleagues who may have missed today's live event. That's all we have for now. Thank you for joining us.